A very good morning to one and all. Respected sir. Sir, we first welcome our chief guest, Dr. Venkat Jasti Garu, Managing Director and CEO of Suven Life Sciences. Welcome, sir.
Thank you.
Sir, did you hear me, sir? So, Dr. Venkat Jasti Garu is the CEO of Suven Life Sciences, and he developed various innovative business models, and he acted as a chairman till 2007 for Pharmaceutical Export Promotion Council, and he is an exclusive statutory body for the promotion of exports of all pharma and biotech products with the support of Indian government. And he is also a chief architect involved in the creation of Pharma City at Vizag with the support of AP state government. So I would like to share your advices as well as feelings towards this program, sir.
Venkat Justice, sir.
Yes, please.
Yeah, you can address the gathering, sir.
Oh, okay. All right.
Your audio is not obtained.
Audio is not clear?
Able to get your audio.
I think you need to admit, I don't know what happened. Can you hear it? Can you hear, please? Sir, can you hear us?
Hello.
Yeah, can you hear us?
Yes,
Yes,
sir.
sir.
Yes,
Yes,
sir.
sir.
Go ahead, sir.
Yes, sir.
Okay, all right. Yeah, thanks for inviting me for this seminar, which is a virtual. As you know, it's a pharmacy profession. You can be whatever you want to in the pharmacy profession. It's a jack of all. Of course, sometimes they say master of none. Is that echo coming from my side or your side?
Yes, sir. There is a little echo from your side, sir.
No, there is small side is running on dual mix.
Huh?
Dual mics పెట్టుకుంటారు side. So
Sir,
So I
it's
think
from our side. You carry on, sir.
maybe you should mute things from your side. Then whenever you need, you cannot hear us, then just unmute and talk to us. Okay. Okay. I'm a graduate from the Andhra University from 1967, 66 to 1972. The idea at that time in the minds of the pharmacist is something different compared to what it is now. It is the testament that your activity, your conference today itself is talking about drug discovery. When we were in the college, our professor, Dr. Subbaravu, used to tell us, after all, you are compounders. That's all it is. Those days, everybody else is great and pharmacist is just a compounder. That's the thought process they have given. But that's not what it is. As I said, it's a jack of all trades. We can be involved in the healthcare systems. Actually, we are the frontline workers in the healthcare system, especially which is evident from now with the pandemic that is raging in. Our profession is a passport to whatever you want to achieve if you put your efforts in the healthcare system. As you know, he may be right when he says a compounder, and the evolution of Coca-Cola, a pharmacist has did the compounding of medication which is not palatable by using this Coca-Cola formulation. We started that way anyway. But we have metamorphosed from just dispensing drugs or distribution of drugs, whatever you may call it, to all the way from drug discovery and the development and the distribution in between, in addition to the healthcare legislation and even prescribing medications. That's what it is. We used to be, those days, even though our professor says it is a compounding of drugs, but our aim is always go to a manufacturing of those days. That's the way we have started. And then afterwards, we went into pure distribution. If you take my case, I started as a manufacturing chemist in Warner in Hyderabad. Then I moved to US. Then there I did the dispensing of the drugs. And that's what we have done. And when I came back to India, I started making the raw materials, APIs to the drugs. And then again, of course, in 2005, I started my own drug discovery program and started in developing drugs for the unmet medical needs. So it's a full circle, as you could see. It is up to you where you want to be. It may not be possible for everyone to take all the steps, but it has to plan properly and do the things right. And as you know, the opportunities are enormous for pharmaceutical people compared to others, which is very much focused on to the task they are set out to do. But when you become a pharmacist, you can become a kind of whether you go into the research, or you go into manufacturing, or you go into the distribution, or you go into public health policy, or even prescribing medications in the dispensing medications in some parts of the country in the US. And authorizing the drugs for distribution and seeing the efficacy of those same the drugs to the distribution. And if you come to the drug discovery, see various activities there where you can play a role in the pharmaceutical chemistry to analytical chemistry to quality assurance to the pharmaceutics. The number of activities you can do. I mean, the N number. It is all what you want to do. You cannot just think that I have only this job is set out for us to do. You can do whatever you want to do, and that is the way this will help not only serve our needs but also help us to take care of the healthcare systems of the world and being a part and parcel of the total healthcare system, maybe the development or in the clinical sciences or the regulatory or the distribution, you name it. This is the way it is. So people should not think that we are going to do only certain part of the activity, but it is all your mindset and your focus and of course your interest. And not all of that will happen in one go, but it will take time, but we have enormous opportunities to do that. And it's very difficult to articulate anything in a remote thing. I think it is better if somebody has any questions, maybe they can ask us rather than giving a lecture which you guys all know what it all about it. But with my lifetime experience and whatever I have achieved or gone through, I can share those things if you have some questions rather than me telling you extempore what is pharmacy all about. With this, I stop, and I also wish this conference a great success. Over to you. Anyone from the podium? Can you hear us?
Okay, sir. Nobody is asking. Thank you, sir. Thank you for spending your valuable time with us.
My pleasure. My pleasure.
Thank you, sir. Now I request MUI Naidu Garu, Director of NATCO Pharma, to the next successive session as a resource person.
Please unmute.
Yes, sir.
Yes,
We
sir.
are
Yes,
able to
sir.
hear you.
This thing is visible to you. Hello? Is it visible to you?
Yes, sir. Yes, sir.
So I can start talking.
Yes, sir.
So good morning to everybody, and I'm
sir.
very much.
Sir, I can introduce you, sir, now.
Hello?
Sir, I will introduce you, sir.
Okay.
Okay, sir. Then we will start the session, sir.
Okay.
Okay, sir. Thank you for accepting our request, sir. So I want to introduce the Dr. MUR Naidu Garu. director of the NATCO Pharma, Hyderabad. So Dr. Naidu Garu has started his career as a lecturer and then elevated as a demonstrated in the Government Medical College for 7 years. And he served the senior research executive in IPDL Research Center, Hyderabad for 6 years. And he is the professor, head of Department of Clinical Pharmacology and Therapeutics at Nizam Institute of Medical Sciences. So actually he established this department in the Nizam Institute of Medical Sciences, and later on he is acted as a currently acting as a director in NATCO Pharma Limited, Hyderabad. And our speaker has organized and delivered 18 international conferences and workshops, and he is the member of International Advisory Board of Indian Pharmacological Society and also a member of Indian Association for Medical Informatics and American Association for the Advancement of Sciences. And he authored a total of 11 books and chapters and developed validated non-invasive sensitive digital pulse plethysmography technique for the evaluation of vasodilators. And our speaker had 79 papers in international and 70 papers in the national journals of good reputed journals. And he is awarded for the excellence in medical profession in 1994 from all the Indian Medicos Society of India. And also he got the poster prize at 5th Asian Regional Course at Dhaka, Bangladesh for promoting rational drug use. And he awarded UK Seth Gold Medal for the best paper in the clinical pharmacology session in IPS 2000. So, sir, you can please give your lecture and valuable words to our participants, sir.
Okay, sir. Thank you very much. And welcome to this presentation. I don't know. As I'm telling you that my presentation will be I'm actually going to talk on the technological advancement and with implication to drug discovery and development. I will be presenting on the following lines. One, I will be talking about the introduction. Then I will be telling about the discovery, advancement in preclinical drug discovery and development. Then I will tell something about a clinical development, which is very, very important, and how the new technologies are working there. Then I will tell about something very, very good, that is organ and human chip technology, which is coming up very much in advanced way. Then I will tell you something about what is known as artificial intelligence and drug discovery. And lastly, then I will go for the coverage of a conclusion. So, now coming to the introduction, actually the drug discovery process and pipeline, they are actually a development process of pipeline, and they are mostly consisting of the various things which is related to, for example, you have got the target selection, then you have got the lead discovery, then you have got medicinal chemistry, then in vitro study, and then and lastly you have got a clinical. So this is actually the drug discovery process or a pipeline. Now, very important issue is that we have to consider actually what is actually the timeline. How actually time takes to develop a drug. For example, if you come to basic research, it takes about 3 years. And after that, you get a target actually identification and selection. Then there is a second thing that again takes 3 years, and the lead discovery actually provides you a candidate selection. And after candidate selection, there is a preclinical development that takes about 1 year and usually 2 years. And then you submit what is known as the investigational new drug filing. Once you do the new drug investigational filing to regulatory department, that they will allow you to go for what is known as clinical development. When you are doing a clinical development, you do phase 1, phase 2, phase 3, and then you submitted a new drug application to regulatory for the marketing. So it takes about maybe 12 to 15 years to develop a drug from basic research to final thing. Now, before actually going the drug we are using for a disease, so what is actually you mean to say that disease is concerned? Now, if you go, disease or the result of distortion of the complex molecular network. It's a very, very difficult thing to understand the disease because it is highly complex and biomolecular network is there. And if you really look into the history of drug development, we find there is a huge gap actually existing in drug discovery and development. Why this is huge gap? The lack of adequate measurement technology limits the process of drug discovery. This is very important. Still, actually, we don't have a very, very good technology to develop a drug. So that is one of the limitations. What is that? We simply do not have the tool to measure the biochemical detail of human body with resolution needed to fully realize the amazing. We are doing it, but we are not having a full resolution of attending and amazing the potential. So this is actually responsible for the drawback for a gap in drug discovery. Now, actually, if you just see a scenario of last 30 years, you will find that there is a tremendous change in the drug discovery program. With the changing the goal, because the goal of the drug discovery, if you take 20 years back and now it is a significant change. And due to that, actually the entire research process is changed. How I will tell you. If you go to 1980 and 90s, the drug discovery was mostly concentrated related to disease symptom. A particular symptom is there and drug is developed for that. We are always considering that all disease is uniform. You take hypertension, we are considering that hypertension is uniform. Everybody is having hypertension, it is same. You can get for, actually you are looking for other disease like depression. All are uniform. Uniform patent. Every patient we are actually thinking is uniform. We are giving only one drug. Universal treatment. Once you diagnose a particular disease like hypertension, start beta blocker. Sickness oriented. All actually disease and drug development are actually related to patient oriented and sickness oriented. This was happening between 80 and 90. Now there is a significant change. What is the change? Now actually we are developing a disease based on the disease mechanism. There is a heterogeneity of disease. Each patient of that disease not necessarily same. Every patient of hypertension not necessarily having the same reason. There is a significant patient variability. Each patient may differ. He may be having diagnosis of cancer or hypertension or actually having depression, but there is a variability. Individualization of the therapy, what is known as personalized medicine, has become. And now we are developing a drug not only to treat the patient to symptoms, but we have to predict whether a particular individual is going to develop a disease, and we are looking. So there is a significant change in the research and development after 2000. Now let us see what are the changes. Now coming to drug discovery and development, the drug discovery process starts with identification of a target. Then we clone and express a protein. Then there is a primary screen. You can do receptor binding. You do primary function and screening. Then you do pharmacodynamic modeling, animal modeling, pharmacokinetics in animal. You do disease modeling depending on your therapeutic area. You produce a particular disease model and then you test, and then you do actually toxicological or further. This is related to drug discovery process. This is the drug discovery process. After the drug is discovered, the next process starts actually development. Preclinical research, compound synthesis purification, animal testing, then we go for phase 1 trial, phase 2 trial, phase 3 trial, and then we submit actually post-marketing phase 4 trial. So this is actually process of drug discovery and drug development. Now, you should be very careful that if you are planning to introduce any drug and want to do a clinical trial, you remember that you have to have answer of some of the questions before going to clinical trial. What are that questions? You should get a clear answer. One, if the compound is safe with animal. If the compound is not safe with animal, better not to go for clinical trial. So you should be very careful that the toxicity and safety evaluation of the animal should be done very, very cautiously, and you should have clear data. Is the compound likely to be effective in human disease? Do potential benefit weigh the risk? So benefit risk ratio you have to check. Always look that the drug should be safe and it should be beneficial. Will the manufacturing process cost effective? We should not think of developing a drug which is very, very expensive, which we cannot manage. Then will the manufacturing process will be able to meet the quality and purity of? These are some of the questions you should remember and you should get an answer before you are going for a clinical trial. What is the future trend? Today, the pharmaceutical industry are facing with challenge of increasing new chemical entity and gaining greater novelty. Now, actually, all industries are getting good number of novelty biological target. Previously, we have got only chemical. Now we have got biological target. Those who are affecting protein, those who are affecting receptor. So this is one of the very, very important actually future trend which pharma industry is facing. There are two very important challenges. As such, there are so many, but two are very important challenges for the pharma industry. What is that? One is that there is an increased throughput of new chemical entity. So many drugs are coming where we have to identify them and do that. Greater novelty of the biological target. I told you that now we are getting a drug which are working on receptor. They are working on particular type of this thing. So there is a greater novelty. So these are two very important challenges which actually we are facing today. Now things have been changed. We have got bullet card. Now we are about this space structure. So there's a lot of change actually happened. What is that change which happened? With advanced technology capability, drug discovery paradigm is totally shifted. If you see the history of last 10 to 20 years, you'll find out there is a lot of shift in paradigm in drug discovery. For that, we can divide the drug actually into three types of drug discovery. One is actually random. Random discovery is nothing, screening a compound using a partial or screen scheming. This is a random screening. You have got some compound, you have got any model, you test it, this is random. The second one is mechanism driven. We know the mechanism of a particular disease, we develop a drug, and this is actually mechanism. And third is the fundamental or basic science discovery. This is mostly done by actually people, those who are working in institutions, they are doing their PhD. There are three types: random discovery, mechanism-driven discovery, and fundamental basic discovery. Now let us see that actually what is going to happen. Now these are the drugs which are developed by random. Actually, this is the time. Example is that the tamoxifen was identified in 1971. It was actually introduced in 92. It took about 21 years. Now coming to mechanism driven, there are so many drugs. I am giving example of fluoxetine and sumatriptan. 1957 it was identified and it was introduced here. So it takes 30 years, 34 to 35 years. Now coming to basic science, drug discovery, erythropoietin was identified in 1950. It was introduced in 1989. It took 39 years. So there is a lot of lag time between identifying a drug and drug is becoming available for the patient. So there is a lot of lag period. Why this lag period is there? What is the reason that why we are taking so much of time to identify a drug and bring it to market? Okay, a single new technology advancement is necessary, definitely, but not always sufficient to lead to innovative pharmaceutical product. What we need, there are so many technologies, we have to convert the technology as that is needed. This is actually today's requirement. What is there present? Collect the data, relate the data and have an outcome. That is actually happening present, but this is not going to help. What actually want is future. You predict the outcome. You say that if I am going to do this one, it is going to happen like that, and then you do the experiment, and then you predict. This is actually the future. You are predicting the outcome based on information of disease, drug, receptor, and then identifying a drug and doing that. This is going to be the future. Now, what is known as a bed to bed. You have to start with what is known as bed. That means the patient who is suffering with the disease, find out the mechanism, find out the molecular studies, do animal studies, and then you have to go for discovery. Identify target, lead search, lead optimization, drug candidate, human studies, and this thing. So that is from the bed we are developing. This is known as what is known as bed to bench, or also known as reverse pharmacology, which is actually now today's trend. Now, what is the challenges to develop a drug for industry? It is very easy to develop a drug for actually acute disease or chronic disease with simple symptoms. For example, blood pressure. If you are developing a drug for hypertension, it is very easy. You develop a drug, take a patient with hypertension, give the drug and record the blood pressure and find out whether blood pressure decreases. You are developing drug for cholesterol. Just to do, if patient is having high cholesterol, give your drug and check. It is very simple endpoint. If there is a very simple endpoint, it is very easy to develop a drug, but it is not always. There is a problem. The new drug requires new technology, new approach. Many diseases for patient stratification staging is required. Now you cannot simply have, we have got a disease, we don't know. We don't know the stratification. We are not having staging. So what is the need? That example, cancer, diabetes, infectious disease, multiple sclerosis, autoimmune disease. It is very, very difficult to identify and find out a drug and develop. It is not like blood pressure. So if you have got patient with cancer developing a drug, it is very, very difficult because we don't have very straight endpoints to do that. What is the opportunities? Ability to unprecedented number of new molecules. Now you are telling that repurposing. We have got hundreds of molecules available for different treatment. We can look for repurposing. Repurposing nothing but trying to identify a new indication for an old drug. Very good example I can give you, that coronavirus. You will find that there are many drugs which are now introduced and in clinical trial are nothing but repurposing. We have not developed any new drug, especially for corona, but existing drug. Hydroxychloroquine, existing drug. Only thing we have done repurposing and using for coronavirus. So repurposing is becoming a very important tool for getting large number of drugs to develop. Product of biotechnology revolution. Now, because of biotechnology, large number of drugs are coming. Biosimilar, antibodies, herbal drug, stem cell. These are all actually opportunity for pharma industry to look to develop a new drug. What is the opportunity? There are so many, but actually I'm telling you that you have to concentrate on three main opportunities. One, that new technology for investigating a complex biological system. I was telling that disease is highly complicated. Now we are looking for new technology to investigate. This is actually first opportunity. Second is that new technology for measuring the effect. There is not a possibility. Your drug may be producing a very small effect. We need a new technology which I can pick up a very small effect, and new technology for predicting the outcome. I should be able to predict the outcome before doing a clinical trial. For that, I need all these new technology. Now let us see what are the technology which are available today for investigating complex biological systems, measuring drug effect, and predicting the outcome for the opportunities. Effective integration of the new technology is the key. So new discovery development needs a integration of all these technology. Now let us see how actually it is going to happen. What are the challenges? Again, there are three challenges. First is that effective acquisition of integration of advanced technology. I told that there are so many advanced technologies there, but it is very important challenge that you have to correctly actually acquire and integrate them. Which technology will be used for drug discovery, you should properly identify. Second thing is that conversion of the data from genomics, other high throughput data gathering technology, medical relevant knowledge, understanding of complex systems, and underlying cell therapy. And third is that the successful application of this knowledge towards productivity. So these are three important challenges which is there for pharma industry to look for new drug discovery. Now, what is the potential today? Again, there are so many, but I am concentrating on three potential. First and foremost, that you have to look for unmet need. There are many drugs for hypertension or so many diseases. You have to search a disease where we don't have very good drugs. One of the very good examples is multiple sclerosis or cancer. And there are actually some treatments that we have not recognized. For example, today you take corona. Other thing is that improve the efficacy of a drug. So patient response therapy is another very important potential. And one of the most important potential for drug discovery is that you have to look that your drug should be safe. There are so many drugs. They are very effective, but unfortunately, we are not able to use because they are highly toxic. And this is one of the very good examples of cancer. Very good anti-cancer drug we are not able to use because patient is dying because of side effect, not because of cancer. So these are actually the very important potential for industry, the efficacy, safety, and unmet need. Now for this, what actually you need, that we need to know the root cause of the disease. If you know the root cause of disease, you can develop. But unfortunately, human cell is highly complex. What is the problem? The human health depends on well functioning of the body assembly. First is the cell. Cell should function properly from DNA to that. All the cells are functioning very well, all the organs will work very well. If all the organs are working very well, the body will be working well. So anything going wrong here, which is affecting cell, will affect an organ, it will affect the body. So you have to understand something here to develop a drug. If we hope to understand the biology, we need to know, looking at one little protein at a time. That is not how biology works. It is very important. You should understand. Simply just knowing a very small protein, that is now how the biology works. What actually you need? we need to understand the integration of thousands of proteins for dynamic challenge for environment. Then only we can correctly develop a drug. Now I will go to the second part after introduction, that what is the technological advancement which is available today with us for the implication of preclinical drug discovery, which is mostly actually before we are going to clinical trial, what are actually the technological advancement which is available to us. There are new platforms. For example, you are talking of in silico, source of new molecule target. Genomics, the great advantage, source of new molecule target on genomic basis can be done. Computational chemistry, this is third, and then high throughput screening. So these are actually our future new platform technology which is going to help us in discovering a drug which is more beneficial. First, coming very small information about what is actually in silico drug discovery. I am not going to cover in detail because already somebody is going to cover it. But very briefly, I will tell you that what actually, why you are looking for a computer-aided design, because besides the significant cost, time associated bringing new drug into market, some major reasons for pharma industry to look for alternative or complicated methods for experimental screening. What are that? That in a survey they have shown that if you are screening 40,000 compound, actually only 5 actually compound goes for animal testing. And out of 5, hardly 1 compound actually goes into clinical trial. You can imagine that you are doing 40,000 screening, getting only 5 compound, and out of 5 you are getting 1. On the other hand, tremendous actually increment in chemical space, target protein receptor, demand of high throughput screening as new lead identification, rational approach, reduce the cost and enhancement of the efficacy. And very, very important contribution is that there is a significant advancement in computer technology of software, hardware. This is actually helping us in identifying molecules very quickly and what is actually what you are talking of in silico. What is in silico? In silico actually is a Latin word, and it is meaning that it is performing using computer or via computer. Actually, Professor Pedro Martínez, this is the individual, is a mathematician from the National Autonomous University of Mexico. He was making a presentation on report of DNA, RNA, physiological content, cellular autonomous and molecular evolution. When he was talking first time, actually in his talk, he used actually in silico. This is the man, actually he brought the term in silico first time in the literature. Now, why there is a need? Why we are looking for in silico technology? What is the advantage? These technology they offer a delivery for candidate more quickly, very fast you can develop, and cost is very low. I will show you that what is the cost of developing a drug by using in silico. They increase the chance of success in many stages of drug delivery process. They facilitate accessing huge amount of data generated, and last, they transform the massive complex biological data into workable knowledge. So this is the reason that we are now actually concentrating on using in silico in drug discovery. This actually what I was telling you, that a typical cost of experiment. Computer modeling only actually $10. Then if you go on biochemical assay cell, gradually it increasing. Once you come into human clinical trial, $500 million. So the cheapest is actually $10 is a computer modeling. So it takes very, very huge amount, maybe from $8 to $12 billion to develop one drug. What is the application? It can be used to analyze the target structure possible, generate the potential molecule, investigate their drug likelihood, log these into molecule and their binding site, rank them according to binding affinity, and lastly, you can optimize molecule to improve the binding characteristics by using in silico. There are some limitations. What is the limitation is that sequence implies the structure for implication. No doubt, the selected protein structure from databases after improving and actually removing various proteins by high sequencing, we get a structured template and alignment. Okay, these alignments are okay, but not necessary that they will work. Second thing that these computational model often represent only fraction of the full length of desired protein. It is not possible that entire protein which is useful as a drug, I can find out with silico. I get only fraction. This is the limitation here. Often drug market price is very high. And one should remember that this is not because of manufacturing sole cost, but price is because of failed drug. I was screening hundreds of compound and lot of money I have invested, that cost is also increased. So screening silico study can help in reducing the production of the cost by, and then market cost also will come down. In silico represent the way format is to spend less for toxicological study, thus save animal to be used in experimental. Third thing, that the real challenge is not to identify a best method to human being environment. This is actually not a real challenge. What is the real challenge? Real challenge is that it takes advantage of contribution from each success. That means you have to use in silico, in vitro, in vivo, which offer the information and develop a drug. This is another thing, is that high throughput screening. Almost 1 lakh of sample, actually within 24 hours, you can screen and you can get your compound. The tremendous advanced technology for drug discovery. So this is actually high throughput screening. What is the advantage? Application, you can do very quickly pharmacokinetics. You can do actually membrane permeability assay. You can find out whether your drug will be absorbed properly or not. You can do actually improvement in solubility, detection of actually cause, whether your drug is going to produce cancer, you can pick up with ISTE. Then mutation screening is possible. Now, actually, I will go to actually the anti-cancer drug discovery. What are actually the latest technology which is helping us? Because cancer is one of the major problems where we are looking for a new drug, and there is a lot of advancement in drug discovery in anti-cancer. Now, what is hot in 2020 in oncology? What is the conventional method? Suppose I have got a drug which for anti-cancer compound. The conventional method is that to do what is known as a xenograph model in nude mice. Here is an example. I am using a lung cancer cell A549, injecting into nude mice. Tumor is developing. I can try my drug. Here is an example. This is actually controlled size of the tumor. This is lapatinib, which is used for, again, the treatment of standard drug. This is my drug M1 and M2. So simply by using actually a nude mice xenograph model, I can find out whether my drug is effective or not. This is a conventional method. You can measure the tumor here and you can quantify. This is the tumor size after resection. This is control. This is lapatinib. This is my drug. It is a very simple method, most commonly used for evaluation of any anti-cancer drug. Here, example, I am giving lung cancer. This is when actually you are developing a drug for brain cancer, that is glioma. You have injected the brain cell into the mouse, and after 30 days, beautiful tumor is developed. You use your drug and see whether the glioma tumor has come down. again you can say a beautiful tumor you can see here by injecting U87 mg. This is nothing but a glioma cell injected into nude mice brain, and the tumor develops. What I am telling that I have to take a mice, inject it, sacrifice. But today I have got a micro CT. I need not to kill the mice. This is the example here. Here is the vehicle, and I do micro CT. I need not to kill the mice, and I can find out. This is the advantage. I am using now micro CT to evaluate my anti-cancer drug for brain cancer, or I can use PET scan for colon cancer. When actually I am developing a colon cancer, I need not to sacrifice the animal. I can do PET scan. So this is actually technological advancement which is helpful for me detecting an anti-cancer drug for colon. What is actually the study in nude mice? Most common method requires nude mice, which is very expensive. One type of cancer only can be detected in one mice. In one mice, I can only inject one type of cancer. Need more amount of test compound, because for mice, I have to give large amount of compound. So, and it takes about 30 days. So no doubt it is very good. But is there any alternative? It is a better alternative as compared to nude mice for a discovery of anti-cancer drug. Now let us see what is actually the alternative available. That is actually known as use of hollow fiber assay for evaluation of anti-cancer drug. This hollow fiber assay is now used by the National Institute USA for evaluation of the drug. How it is done? Actually, we take these hollow fibers, these are 2 centimeter fiber. They are available in different colors. Here I am showing blue and yellow. Then there is actually orange or red. What you take? You take 2 centimeter of this hollow fiber and put your cancer cell into that and fuse it. Now this is ready-made cancer you have prepared on packaged it in the form of hollow fiber. You just put into mice. Now this tumor is ready in the form of hollow fiber, and any cancer cell you can put into that. And what you do, just example here, I have taken 2 cancer cells. One is blue, one is yellow. After 10 days, I can take out and I can find out what is the amount of cancer cell in there. Yeah. Another advantage is that when actually you are using hollow fiber, you can also see that growth of blood vessels, what is known as angiogenesis. And we know that if there is a more angiogenesis, the tumor will be spreading to other tissue and metastasis is there. You can also identify whether your drug is preventing angiogenesis. If your drug is preventing angiogenesis, it will also prevent metastasis. So Fiber becomes a very simple and very good model for evaluation of anti-cancer drug. You can have imaging facilities. You need not to sacrifice the animal. Here again, I have put hollow fiber with luminescent imaging, and after 2 to 3 days, I am using a microscope and I can imagine and find out the fluorescence. Here, actually, orthotopic model of breast cancer using luciferase tagging. Just I can identify and I can find out the tagging luciferase in vivo imaging in a tumor which is there. This is an example where I can actually evaluate a anti-leukemia drug, injecting the leukemic cell directly into the vein of the mice, and beautifully that this is actually the days, and then you can say as the days are increasing, beautiful leukemia development I can quantify. I can give the treatment which is anti-leukemic, and I can find out whether this fluorescence is coming down. This model is becoming very simple, very easy for evaluation of anti-cancer drug, either anti-leukemia drug, anti-colon, or glioma cell. What is actually advantage of hollow fiber? Why we wanted to do hollow fiber? Actually, in one mice, I can try 4 types of cancer cell. I can use 4 actually hollow fiber, one with the breast cancer, other with the lung cancer, other with the pancreatic cancer, other with the colon cancer, only in one mice. And within 7 to 10 days, I get my data whether my drug is active or not. I can check angiogenesis, and it is very cost effective. Only 5 mice, I can evaluate all my drugs in 4 tumors. So this is a great advantage of using hollow fiber assay method for anti-cancer drug. Now there is something more hot in oncology, that is actually role of zebrafish in anti-cancer. This is a zebrafish, and zebrafish has now become a very important tool for drug discovery and development. I'm only going to concentrate on anti-cancer. This is the individual Dr. Thomas Cook. He was explicitly using zebrafish for evaluation of various anti-cancer drugs for leukemia and solid in actually Dana-Farber Cancer Institute in USA. Why hot is that this tiny vertebrate belonging to a beneficial player in discovery. Why actually zebrafish is becoming so popular? Most platforms in drug discovery are either in vitro assay based on the cell or part of cell, or in vivo assay based on vertebrate. Both set are with pros and cons. So presently what you are using, actually you are using in vitro assay or in vivo assay in mouse. And so what actually advantage that when you are using zebrafish platform have a potential of combination advantage of both in vitro and in vivo. I will tell you how it is happening. Why zebrafish model system is becoming well popular? It is very small. It has got that. This is a 2 type of, one is female, that is very small size, very short life, generation time is very quick, and very quickly you can get as many as zebrafish you want in your lab for use. Good reproduction, large number of, if you take one female, one male, you'll find hundreds of zebrafish you can develop. External fertilization, you can see the actually embryo, ova and larva. It is totally opaque. You can see that all organs of the zebrafish you can check. Embryo is totally transparent. Rapid embryo development. So these are actually reasons that we are preferring now zebrafish as a model for anti-cancer drug. This is the embryo. Now this embryo has become very popular for evaluation of actually safety of the drug. Here now where OECD guideline 236 has come, what is known as fixed embryo acute toxicity test. Now this test has become, you can very easily check whether your molecule is safe or not in actually zebrafish embryo. I'll give an example here. Actually, this is 3,4-dichloroethylene, which is standard drug you can use, and this is a normal control. This is actually a embryo injected control at different dosage. And clearly you can see that when you are using 2, 4 or 8 milligram, you are clearly developing coagulation. So the toxicity of your molecule, you can very quickly find out in actually zebrafish embryo within 96 days. Safety evaluation has become, and we have got the advantage that this particular guideline is approved by OECD. So you can use actually zebrafish embryo for screening of your compound as far as safety is concerned. This is actually adult zebrafish. All organs you can see here. This is a fluorescent microscope, because when you inject any cell, that cell should have fluorescence. Example here, we have injected a green fluorescence, actually protein containing glioma cell into zebrafish, and nice actually fluorescence is there. You can quantify this fluorescence. Example here, after 2 days, this is the vehicle. You can see the size of fluorescence. And when I'm using my drug, which is very good for glioma, there is a significant decrease. I can quantify this fluorescence. Zebrafish becomes a very good model for me to test anti-cancer drug for glioma. Within 3 to 4 days, actually, I get my data by zebrafish. Now, after the preclinical, now let us go. What is the actually technological advancement which is available for clinical evaluation, phase 1 trial, phase 2 trial? When you are going in healthy subject or in patient, what is the technology which is available for drug discovery and development? All of you will agree that pain probably the most common universal condition, and all the people, those who are actually attending this webinar, I know that sometime in their life they might have actually pain and they might have required the analgesic. You can see that $50 billion are spent every year only for back pain in America. So large number of people are using painkiller, and for industry, it is actually a big boon. Suppose you are developing a very good painkiller, you are going to have lot of money. So actually pain requires a very effective analgesic. So industry is looking for a good analgesic agent market. Now what is situation today? I'm just telling, seeing is believing. I think you all will agree, yes? Now the question, can we see the pain? Anybody can answer? No. So I will tell you that what is actually, let us find out the answer. Can we see the pain? Because pain is very important for me to develop a drug. If I can see, I can do. What is the conventional method? Suppose I have got analgesic drug. I have to take a patient who is suffering with headache, or there's a tooth extraction pain, or post-operative pain. What is the limitation? That response is highly subjective. I have to only depend on patient to tell whether patient has got relief with the pain, how much pain relief is there. Inter-subject variation is very high. Large number of patient is required. So there is a lot of limitation. I have to depend on the patient. Large number of patients are required. It will take a very long time to develop a new analgesic drug. This is a conventional method. Are we having something where I can develop my drug and find out whether my drug is good analgesic or not very quickly? Now let us see how. Now I'm talking of a technology like the molecular imaging, that the PET scan, function MRI, genomic mapping, all these technology which are available, they are highly sensitive, highly specific method. And what is the application here? If I combine all these technology which is available today to me, I can study disease, I can study function, I can do targeting, pathway, structure, function, organ, tissue, receptor, cellular, and genomic. So this technology has really significantly contributed to me for new discovery and development. Now let us see how they are actually becoming useful. I will take, for example, of MRI. You might have seen this MRI in many hospitals. It is there. But this MRI, if you are there, MRI only provide information about the structure of a brain or a structure of a particular organ. We are not interested in MRI, which is only giving structure. As a pharmacologist or clinical pharmacologist, we want actually, we don't want to see how brain tumor is looking, whether there is a clot or there is a bleeding. This is only for doctor or surgeon. As a pharmacologist or as a clinical pharmacologist, we are more interested in knowing the function. And for that, I have got function MRI, and function of the brain or function of particular organ I want to evaluate, I need function MRI. Now this function MRI is available for us for evaluation of new drug. Now let us see about pain itself. Now this is actually functional MRI setup. Potential application of functional MRI in drug discovery, I can stratify patient. I can find out which patient is having this type of problem. I can do all pharmacodynamic response. I can check with function MRI. proof of mechanism I can do. I'll give an example here. Early phase outcome, very quickly using functional MRI, I can pick up whether my drug is acting on particular disease or not. Surrogate marker outcome. Now I'll give example of using functional MRI for experimental drug. I've got a new analgesic drug I want to evaluate. On actually healthy subject, I use a laser beam to induce pain, and I will also ask him to score here. So when actually laser beam is given, the individual may be healthy subject getting a pain. When he's getting a pain, I use a functional MRI. When I do use a functional MRI, I get a brain mapping. And by my knowledge of medicine, I know that the area in the brain must actually perceive the pain. When there is a pain, you can see there is a large increase in the intensity of that area of thalamus, and I can quantify. So during pain, thalamus becomes very hot and you get a hot spot, and you can quantify by functional MRI. Now I have got a drug, fentanyl, which is a very good analgesic drug. When I use fentanyl and then induce pain, you can see here that there is no change. Simply using functional MRI in 20 healthy subjects, I can find out whether my new drug is good analgesic or not. But functional MRI is not available in most of the places. What we did actually, only thing I have to record the blood flow in the thalamus area. We started actually using cerebral blood flow by using gamma scintigraphy in the nuclear medicine department. This is a gamma camera. And the experimental pain SPECT model. What we did actually using pain, we have used actually a bottle cap we put onto the volunteer's forearm. On that, we have wrapped the BP apparatus cup, and gradually you increase the BP pressure to 100 to 200 millimeter. And when there is a pressure pain, individual gets severe pain. When he's getting a severe pain, you inject a radioisotope that is nothing but tritium-99. The tritium-99 will go into brain, and wherever there is an increase in the intensity of pain, it will be able to find out, and you can take a picture and map. For example, here, this is the picture of gamma scintigraphy. When there is no pain, we are not able to get anything. But when actually the participant is getting severe pain, we are getting, and you can quantify. Gamma scintigraphy was used for evaluation of an analgesic drug in healthy subjects, and within 1 month you can get your data whether it is effective or not. There is another very big problem. Suppose you want to develop and find out a drug for mental disorder like psychosis, schizophrenia, dementia. It is a very difficult task. Very, very difficult to do a clinical study and pick up a new drug for any psychopharmacological problem. Why? The reason is that there is a lot of limitation. What is that? Very difficult to diagnose a patient whether he's suffering with schizophrenia, dementia, or because there is a lot of overlapping. Same patient is having depression and then he may go to anxiety. This is one limitation. Usually, we use a visual analog scale, mini-mental scale to evaluate a psychotropic drug for actually this thing. This is all subjective. High subjective variability. Patient to patient variability is very, very high. And for that, you need large number of patients. 300 to 400 patients you need if you want to pick up your drug, whether it is as a psychopharmacological agent for psychosis or schizophrenia. So this is one of the major limitations. Very difficult to identify a new drug for actually dementia or schizophrenia. How this limitation is there? Now we have got again a function MRI. When you do a function MRI, a patient with schizophrenia, you get a typical site where actually you get a mapping. In case of psychosis, you get a typical mapping on particular brain area. In overlapping, both of them. So using functional MRI imaging, we can pick up patient is schizophrenia, psychosis, or overlapping syndrome. Before drug, you identify the quantification, your drug, and then you again do functional MRI, and very nicely you can quantify the effect of your drug. Functional MRI imaging has become a very important tool for evaluation of drug for schizophrenia or psychosis. What is other thing? Again, thing is that we are knowing that the benzodiazepines are there. Example here, the lumazenil, which is actually having effect on GABA receptor. Today, using SPECT, I can actually find out the GABA receptor in the brain. I can quantify. I can use my drug and I can find out whether my drug is occupying a GABA receptor or not. And if it is there, then definitely my drug will be there. So using SPECT as a technology, I can evaluate whether my drug is acting on GABA receptor very quickly, and I can go ahead with a clinical trial. Other thing is that actually SPECT I'm able to use in patient with schizophrenia using clozapine. Clozapine is a drug which is useful for a treatment of schizophrenia. Here you can see that individual patient not treated, large number of actually area I'm getting in this cerebral distribution of dopaminergic receptor. This is when there is a complete blockade when you use clozapine. There is a partial blockade. By using a SPECT, actually I can find out whether a schizophrenic patient is responding with my new drug very similar to clozapine. This technology has become very handy for evaluation of drug for schizophrenia. Now I will tell you something very, very important, that a psychotic eyeball movement recording and determination of what is known as the peak psychotic velocity. Why? Because it seems actually this particular peak psychotic velocity recording, it is very effective, very selective for evaluation of benzodiazepine. If you have got a new benzodiazepine, if you use this actually peak psychotic velocity recording, you can evaluate your drug. I'll tell you how. This is actually the recording setup where actually you have got the diode here. These diodes are placed at actually 10-degree distribution, and there the individual, he may be healthy subject, he has to concentrate on these diodes, and when these diodes are lightning, they have to move the eyeball very fast to these diodes. Example here, we have got the electroocular graph and close recording the eyeball movement. This is the example where we are recording eyeball movement. Again, you see here, these are the lightning. This is with each 10-degree on to the right side, and then actually the same thing will happen on the right side. And this is with the 10-degree difference. Individual healthy subject has to just move his eyeball whenever there is a lightning happens. And with the movement of eyeball, which is there, record the eyeball movement like that. With 10 degrees this much, 20 degrees this much, like that. With the change in the degree, you can find out there's a change in the movement of eyeball. And with the computer algorithm, I can find out the duration with each time interval, and I can find out the speed. So displacement, duration, and the peak velocity I can find out. If I can quantify, I can check my drug like benzodiazepine. Example is we have used temazepam as actually you can see this is the concentration of temazepam. When the concentration is going up, you can clearly see that the peak actually psychotic velocity was coming down, and actually this is the concentration as compared to control. So using actually peak psychotic velocity, you can use it for picking up a drug like tapazolam because it is very specific for benzodiazepine, and we are very much thankful to Biomedical Engineering Department of Osmania University for giving us this particular model for evaluation. These are some of the publications. Now I will come to something very important, that is the actually positron emission tomography, that is PET scan. This has become very, very important, very good tool for evaluation of drug discovery because it is highly versatile for drug discovery development. I'll give example how PET scan is useful. What is the role of PET scan? Actually, I told you that we have got CT MRI, it only gives the structure. I'm not interested. I'm interested in the function. What actually I get? The actually PET scan gives physiology, metabolism, drug distribution, molecular pathway, receptor, highly sensitive, very specific, and this is. So wherever actually PET scan is there, all these things actually I can evaluate, and which is very sensitive, very specific. So PET scan has become a very important tool for early drug discovery. Now let us see the example. We can study tissue metabolism, blood flow, functional, pharmacodynamic, pharmacokinetic by using PET scan. Here is the example. I want to check the distribution of drug into the lungs. We know that triamcinolone is used for the treatment of bronchial asthma. When the individual is not using spacer, you can find out the distribution by PET scan. When the same individual is using spacer, the amount of actually the triamcinolone going into lungs is very high. Using PET scan, I could quantify the distribution of the drug. This becomes a very important tool for me to study the distribution. That's the reason now we are telling that if the patient is using inhaler in bronchial asthma, you must use space because the distribution of drug is very high. That is possible by PET scan. So distribution of drug, I can very easily find out with doing a PET scan. Now, this has become a very important thing for evaluation of anti-cancer drug. PET scan, this is before treatment, I can find out both in the liver and in the urinary bladder how much actually the thing is there, and after treatment. So using FTG PET scan, I can evaluate an anti-cancer drug for evaluation, whether it is effective, what is the dose required. Now I will come to another condition that is actually migraine. Very common. Again, conventional method needs a visual analog scale and so many limitations are there. What is actually, we know by our actually knowledge that in case of migraine, actually the patient develop severe extracranial vasodilatation and temporal pulsatile pain. So there is a severe pain which is pulsatile. And our knowledge of pharmacology says that if you use actually nitroglycerin, one of the side effects of nitroglycerin is actually vascular headache, very similar to migraine. So this our knowledge of actually inducing headache because of nitroglycerin, we can use as a model in healthy volunteer. What we have to use? We have to use experimental setup where I can actually quantify the blood flow in the extracranial vessel before and after nitroglycerin. So this is the example here. Sumatriptan is a drug. When I'm only using sumatriptan, this is the percentage change in the blood volume. It is not there. When actually I'm using nitroglycerin, there is a significant increase in the volume. When actually I'm using sumatriptan with nitroglycerin, it is blocked. When I'm using my drug, again with nitroglycerin, there is a significant decrease. So clearly shows that the volume of blood flow, which is increased in the temporal vessel, was significantly decreased by my drug. Now, second parameter is that what about the caliber of vessel? Again here, you can very clearly see that when I'm using nitroglycerin, there is a vasodilatation and my drug afterwards is completely blocked. So this new investigational product may be a very good drug, very similar to sumatriptan for the treatment. What we do, we also use a visual analog scale to find out the headache score, and we have seen the headache scale. Again, you see only nitroglycerin, lot of headache. When that you are using, the headache also has come down. So this actually model is very simple to evaluate drugs like sumatriptan in healthy subject. There is another great challenge for multiple sclerosis, very difficult. Why? Because once you diagnose, the life expectancy of the patient is only 5 to 10 years. We don't have any drug which is very good for multiple sclerosis, very difficult to diagnose. Multiple sclerosis becomes a very important indication for developing a drug, but lot of problems are there for diagnosis. Again, here functional MRI has become a very important tool because in multiple sclerosis, there is actually relapsing. Sometimes symptoms are more, sometimes symptoms are coming down, and throughout the year, this will happen. And what are the changes which will happen? One thing is that, number one, the brain volume usually comes down. Number two, there is inflammation. These yellow bars are inflammation. And number three, there is axon loss. So when the patient is developing multiple sclerosis, gradually there is a decrease in the blood volume, the brain volume, there is inflammation, there is axon. Today, by using functional MRI, actually I can quantify this. And if I can quantify this, how I can use? Here is the example. Actually, you do a early evaluation. Here example is there when you are using placebo. This is healthy subject, and this is actually abnormal reaction you are getting it. And these are actually patients of multiple sclerosis. You can see that their actually reaction index are very high as compared to the healthy subject when they are on placebo. When you have got a drug which is useful for multiple sclerosis, like rivastatin, all actually when you do, the activation ratio has significantly come down. It was very high, it has come down to almost normal level. And this activation ratio can be quantified by doing functional MRI. Functional MRI has become a very important tool today for evaluating a drug for multiple sclerosis. This is something very bad. They are telling 2025 India is going to become a capital of the world for diabetes complication, like vasculopathy, coronary artery disease, stroke. I highlighted neuropathy. Why? Because it has got something special to do with diabetes and drug discovery. Diabetic neuropathy, what is the major complication? There will be gangrene, and finally, actually, the consequence, you have to cut the leg, so amputation, because this is one of the major problem in diabetes. They develop gangrene and the individual has to lose. This is only because of what is known as a neuropathy. Now, early detection of neuropathy, if we have got something, we can actually detect a complication of diabetes and develop a drug. Now, let us see how. Actually, our peripheral nerves, they actually transmit four sensations. They transmit sensation of touch. When you touch your actually toe, you can feel, you get temperature, whether it's cold or hot, you get pain, and then there is a vibration. So we have got four sensations: touch, temperature, pain, and vibration, which is transmitted from peripheral nerves. Now, what is important? Importance is that in case of diabetic neuropathy, the first sensation which is lost is vibration. Then only pain, then only temperature. So if you can detect very early loss of vibration, and here if you can do something, you can prevent further damage, and there is no loss of pain, low temperature, and patient will not develop gangrene because you are detecting very early. So what you need something where you can actually get a vibration loss very early in diabetic neuropathy. How it is possible? Today we have got the equipment that is actually a neuroaccelerometer, which actually picks up a loss of vibration in very early stage. Example here, we are doing a study where you are using the vibration actually potential threshold. This is known as the VPT. It is directly related with the loss of vibration threshold. We did a study with using gamma linoleic acid. It has been shown to be very effective in the treatment of diabetic neuropathy. We have done evaluation of VPT in normal subject and diabetic subject. You can see here that there's a very high actually change in the VPT as compared to the healthy subject. So many of the diabetic patients, they are having diabetic neuropathy so high. And when we treated these patients, 52 patients, and we find that there's a marked decrease in their. So actually, the VPT becomes a very important tool for detecting of early diabetic neuropathy. And if you have got a drug, you can prevent diabetic neuropathy and you can prevent the limb loss. Then psychomotor performance, I told you that there are various methods. We have got actually traditional method and computer method. What I'm going to talk that now the computerized method has become available. This is the computerized method we developed here, and I can show that all these tests you can use the computer method for psychopharmacological evaluation of drug. This is the example where this is the latest version of our computerized psychomotor test, that is Mindomatics. You can evaluate all psychopharmacological drugs very quickly in the healthy subject, very effectively. These are some of our papers which we have published using Mindomatics. Now I will come to something very serious, that what is actually the method evaluation for function of cardiovascular system. Why? Oh, sorry, I think this is the wrong slide has come. No, but I will utilize it for something. This slide, actually this you might have seen when you go to some shopping complex or mall, they mention buy one, get two free or three free. This is attraction. I will give you a better option. What is that better option? Buy one, I will get you 8 free. What is that? What you have to buy? You just buy diabetes or obesity. You actually develop obesity and you will get all 8 things like hypertension, diabetes, all this will be free. This is the complication of obesity. Simply developing obesity is more than enough for you to get all complications. Here I am talking of hypertension, diabetes, and heart disease because concentrating on cardiovascular. Now, as per WHO source, by 2025, the maximum actually mortality, morbidity is going to happen because of cardiovascular disease. So it is becoming a very serious problem in coming 5 years. This is a very, very important slide of mine. This is actually published by London et al. In nephron dialysis in 2002. What they have done, actually, they have evaluated the patient with chronic renal failure or end-stage renal disease. Along with so many other parameters, they recorded mean blood pressure and pulse wave velocity. What you can see here, that they followed for 5 years. And all the patients with end-stage renal disease, those who survived for 5 years, in them, along with decrease in the mean blood pressure, there is a very good decrease in the pulse wave velocity. But unfortunately, in non-survivors, most of the people, those who died within 5 years, no doubt, the mean blood pressure was very nicely decreased, but actually pulse wave velocity increased. So there is a difference that decrease in the mean pressure is not necessary that it is going to prevent morbidity because there is an increase in the pulse wave velocity. And the pulse wave velocity reflects what? Arterial stiffness. No doubt blood pressure has come down, but if the arterial stiffness is high, morbidity and mortality of cardiovascular risk will be very high. So this becomes a very important actually criteria for evaluation. How? Now there are various methods to record, like I'm talking of pulse wave velocity because the example which I'm giving is the pulse wave velocity. There are various instruments. There is an instrument known as the sigmocar, where you use a radial pulse recording here. you get an augmentation index, and then you can have a radial pulse recording by finding out the center pressure and artery stiffness. You have got ankle brachial index machine. You can find out the flow mediated recording. It actually also gives you an idea of the pulse wave velocity and endothelial dysfunction. What actually I'm going to talk to you about the equipment which we have developed with the help of actually medical genesis system available in Hyderabad. We developed an instrument known as periscope. What actually periscope does, that simultaneously it records the pressure from all the vessels, both the arms and both the legs, and it does the envelope, and it calculates all your actually pulse wave velocity in all the limbs. This is the example. We have method validation which we have published, and we have clearly shown that our method is working very well, and it has got very good correlation in recording the pulse wave velocity and artery stiffness. Here I will give you the example. This is a healthy subject. You can see here that actually there is a significant gap between the y-axis and development of this envelope. Because the healthy individual is having a normal pulse wave velocity, there is a normal artery stiffness. But if you take a patient with hypertension, you can see the loop. Since beginning, there is a huge amount of pressure development. Diabetes, coronary artery disease. So these hypertension, coronary artery disease, diabetic patients, they have got huge amount of increase in pulse wave velocity, which is clearly detected by periscope. Now see the quantification. This is the quantification of carotid femoral pulse wave velocity. This is healthy subject. This is coronary artery disease, diabetes, chronic renal failure, rheumatoid arthritis. All these patients, there is an increase in the velocity and there is an increase in artery stiffness. There is a high risk of cardiovascular risk. Then brachial index also we have done same instrument. Pulse wave velocity in end-stage renal disease. Very clearly we have shown there is a marked increase as compared to control. Now there is another organ, that is endothelium. Now endothelium is not a simple structure, it is an organ because endothelial dysfunction is a very early marker of cardiovascular disease. They say that actually frank cardiovascular disease develops and before 5 years of age, endothelial dysfunction can happen. So if you detect endothelial dysfunction very early and treat, you can prevent most of the complication of cardiovascular disease. How it is possible? Again, this is the endothelial dysfunction, and I can tell you what is the consequences. So many diseases actually has got endothelial dysfunction. Endothelial dysfunction is very early marker. All these people, they develop cardiovascular risk and they die because of cardiovascular, because of early change in this thing. So endothelial dysfunction determination becomes a very important tool. How we do that? There are various methods. I will talk about a pulse wave analysis, which we have done. We have again developed an instrument that is known as a dicrovin. The dicrovin, actually our principle is that you ask the individual to inhale salbutamol, and salbutamol is known to cause release of nitric oxide, which is normally in the arterial endothelial. The endothelial actually having a normal nitric oxide is released and you get a vasodilatation. Simply inhalation of 400 microgram of salbutamol will cause vasodilatation, and this vasodilatation you can pick up. So there is an inhalation of salbutamol, you get a very good vasodilatation. This is reflected in the form of change in B by A ratio, which we have quantified with our equipment. This is salbutamol nebulization. And this is therapeutic. So using salbutamol inhalation, and we have clearly demonstrated, this is healthy subject, normal endothelial function, coronary artery disease, peripheral vascular disease, diabetes, all these patients, there is a clear-cut endothelial dysfunction, which we can pick up. We have used salbutamol inhalation, and we have clearly shown that this is normal and this is diabetic patient. significant. Other thing we have changed actually reflex index, even patient with SLE, very clearly we have shown. So in SLE patient, diabetes patient, even patient with actually SLE, we have used the drugs like statin and actually L-arginine. L-arginine is a substitute for nitric oxide, and statin is a very good drug which improves endothelial function. We have clearly demonstrated that when we treated patient with SLE with L-arginine or statin, there is a significant improvement in endothelial function. These drugs can be used as a good drug to improve the endothelial function, and they can prevent cardiovascular complications. Same in chronic renal failure, we have used allopurinol, and we have shown that before treatment, after treatment, there is a very significant improvement in arterial stiffness and endothelial function in patients with chronic renal failure. Now, one of the very important things that you have to understand that as per regulatory requirement, if you are developing antipsychotic drug or developing antihistaminic drug or prokinetic or antibiotic, you have to prove cardiac safety. Now, there are various methods. One of the very good method for evaluation of the cardiac safety is Holter monitor. This is a Holter monitor where actually you can record actually QT prolongation, and if your drug is cardiotoxic, you can pick up. Here is the example. When we recorded the cardio monitoring in one of our experiments, we found that one participant was developing a very early cardiac arrhythmia, which we picked up in Holter monitoring with phase 1 trial testing a new drug. Again, extra cardiac extrasound. So the Holter monitor becomes a very important tool. Coming to blood pressure, when you are developing antihypertensive drug, you have to record 24 hours blood pressure, which is not possible by simple sphygmomanometer. You need an ambulatory BP monitor. Record day and night, and then you can record a complete 24 hours blood. When your drug is acting, how long your drug is acting, what is the peak effect. It is only possible if you are using a actually ambulatory BP monitor. And as I have told you that this is one of the drugs which has become available. It is the first antibiotic which we have done phase 1. We have done a cardiac safety evaluation by these two methods. And this is the first drug got permission marketing in 2019 from WOCAD. Now, another very important issue which comes is the compliance of the patient in clinical research. You have given 60 tablets to patient to take one tablet morning, one tablet evening. Whether he had taken or not, we don't know. In clinical research, it is important that there should be 80% actually compliance, then only your data is valid. What is there? There is a model where actually you put all your drug into that, and whenever the individual is opening, that is recorded here. And when he comes back to you with the actually computer algorithm, you can find out at what time patient or subject has removed it and what time is there come there. Now I will come to a very, very important and very recent technology, that is organ and human on chip technology for discovery of drug. What is organ and chip technology? It is actually a multi-channel 3D microfluidic cell culture chip which stimulate activity, mechanism, physiology, response of entire organ. It simulates exactly organ-like thing it is there in a chip. These micro devices are actually translucent. You can see they provide a window for watching inner working of human organ. You cannot see the kidney of the human being, but actually you can see this kidney outside and find out. This is actually the technology today which is available, that is organ on chip. What is the application of organ on chip? This technology being used for development of whole population of the human organ on chip, such as we have got lung, gut, liver, heart, kidney, you name any organ, we have got actually chip. Even I can check blood-brain barrier of my drug on chip. The idea is to recreate a small functional unit any particular organ in microenvironment very closely actually imitating human body. So these are the chips now available for testing. I need a very small amount of drug to test. This is actually lung on chip. I can evaluate my drug for bronchial asthma. Heart on chip. I can evaluate my drug whether it is showing any effect on heart. Artery, I can use it. Placenta, mostly actually in pregnancy, whether my drug is crossing and causing any abnormality in placenta, I can check by placenta on chip. I have got chip on gut. I can evaluate drug acting on gastrointestinal system. This is something very important. All the liver function which is affected by with my new drug, I can check with chip on liver. Then on kidney function, I can check. I can evaluate anti-cancer drug by using actually tumor on chip. How these organ pipe pharmaceutical industry, why actually pharma industry is putting a lot of effort to do that and looking for this new technology. What is the advantage? They are telling that it will replace animal model, but I don't know whether it is going to happen or not, but they are predicting. They will help the laboratory to reach a clinical trial very fast. They are telling within very short time you will reach to clinical trial using organ on chip. Comparison side effect of human and animal, it is possible. Study interaction of pathogen and organ cell, it is possible. And also mechanism of some viral attack, it is possible today. I'll give some example. Study the effect of main action site, whether your drug is acting on kidney, liver, where it is acting, you can pick up by this technology. Study of toxicity of drug. I told you that safety of the molecule you can use in using organ on chip. Study about the cancer cell and you can produce a new drug for cancer treatment. It ensures the regulatory decision. You can actually carry this chip and you can show to the regulator and they can see the effect of this. So you can carry the chip. You cannot carry the organ of a human being who is actually relieved, but you can carry this chip and you can show to the regulatory authority to show that how your drug has improved. And you can develop vaccine and counter bioterrorism threat. These are some of the advantages pharma industry is looking for organ and chip. There are some disadvantages. What are that? Early property of organ cannot be measured. Sometimes it is not possible to study a complete organ, and very important thing that we cannot evaluate using any drug during the pregnancy. These are some limitation of organ on chip. Now let us come to what is another thing, that we have got a complete human being on chip. I've got a man on chip. I can study. This technology is there where I can have all in vitro tests. I can do all pharmacokinetic, pharmacodynamic, toxicokinetic. These all models are possible by using actually human in chip. What is that? This is the chip, which is a microchip. It's a highly sensitive. And I can use this chip and I can inject into the circulation. Once I inject into circulation, this chip is going and circulating into body of the human being. And what I can do, this is the example. I have injected, it is going, it is circulating into the entire body. Now when it is circulating into entire body, what I can get? I can actually get where actually particular disease is locating. I can find out actually disease state, whether disease is increasing or decreasing. I can actually compute the dynamic response, whether my drug is really working, not working. I can find out actually trigger response. I've given some drug to work on particular receptor. I can pick up whether it is going and stimulating or inhibiting that response. And very, very important thing is that I can find out the safety of an organ by using actually the chip which is circulating. So this technology is also becoming very, very useful for evaluation of drug, which can be done very quickly, very fast. This is actually the design year which is given to this gentleman who got actually the award for developing organ and human chip in Harvard University. Why actually this technology would have more pharmacological study, will be used for study, concentration, time effect, drug effect you can do, will also help to pinpoint whether those are drug both effective or safer. Ultimately, organ on chip innovative technology would help develop the new drug discovery very fast. And lastly, it is going to revolutionize the pharmacy field in the future. And they are telling, I think, coming 5 years, all this technology will be becoming more popular and we will not be using actually a healthy subject or the animal for testing our drug. Let us see what is going to happen in 2025. Now I will come to a last topic, that is artificial intelligence in drug discovery. Really, it has significantly contributed. And you talk of any area, it is there. For example, actually they were resolution complete thing is that actually they have found a very potent kinase inhibitor and they can actually develop within 46 days by artificial intelligence. It was taking very long time, but by using artificial intelligence within 46 days, actually they can find out whether drug is showing efficacy or not. Such a fast way, which was taking 4 years or 5 years, they could find out the drug. This is something very great use of AI. Another thing is that a drug candidate which is there, which is very quickly identified by that. Research in this thing is that can predict which research translate into clinical trial. It is possible that by artificial intelligence, I can predict when I am doing such a such thing, whether I'm going to get a particular result or not before actually doing clinical trial. Today it is possible for me. I have got advantage that I can actually predict actually the breast cancer by new AI system. And some of the hospitals are using, they can predict whether there will be improvement or risk by only using AI. Cancer diagnosis, very quickly it is possible by using artificial intelligence. This is something very important. Now they are telling you need not go and give your blood for blood sugar level. No prick is required. They have developed a technology by just reading actually electrocardiogram, a autonomic detection of hypoglycemia by few ECG beats. This is relevant because ECG can detect any circumstances including sleep. They are telling that you can analyze sleep, you can analyze actually your blood sugar level by using artificial intelligence which is recorded to this model. And they have done this study in 80 people and they found that the data is very much correlating by their artificial intelligence. So they are telling it may be going to replace a pick of the tablet for using a blood sugar level. So you can imagine that how much artificial intelligence is going to help to drug discovery and to the patient. Neurological disease, very difficult. Now we have got actually artificial intelligence-based blood tests to predict degenerative disease. I can now predict all degenerative disease like Alzheimer's disease or multiple sclerosis or other type of disease simply by using a blood test and applying artificial intelligence and an algorithm. In cancer study, really it has significantly contributed that simply using artificial intelligence, very quickly I can predict the type of cancer, stage of cancer today. Coming to brain cancer, artificial intelligence algorithm very accurately identify brain tumor and reveal surgery. I need not to go for actually brain CT or functional MRI. Simply by using AI algorithm very accurately, they have found that it is as accurate as 95% to detect a brain tumor. You are telling that smartphone, we are going to have testing of your DNA. All your actually DNA testing data will be now available on your actually smartphone. Coming to vaccine, actually one of the computer generated flu vaccine enters into clinical trial. This is the first example of use of artificial intelligence developing vaccine. I am hoping that same should also happen with coronavirus. Let us see how much, but definitely artificial intelligence is playing a very, very, very important role for drug discovery. Especially for the paralytic people, it is very significant. Now they can use their hand very effectively by using artificial intelligence, and they can operate nicely. All paralytic people, they can use actually AI globe. All stroke patient, now they can work. The man actually who is there, who mostly contributed, is nothing but an Indian, that is actually Sundar Panchayat, which was responsible for many. He's got a very good video. Because of no time, I'm not able to show that video talking about the role of artificial intelligence in drug discovery and patient management. There are so many industries. This is about 3 years back. At times, 100 industries are actually working on artificial intelligence all over the world. And these are the people, and this individual actually is the panchayat. So there are many people. Now they have told more than 600 actually people they are working on, and these are the top 100 leaders 3 years back. Because this is a very popular and very quick and very useful technology, many people are concentrating on artificial intelligence, on drug discovery, not only on drug discovery, many area of human. For human being, I think this is one of the very, very important things which is happening. I think this is my concluding slide. At the end of my career, I can look back and know what something I did made a difference. One patient somewhere in the world, that will be more satisfying and more gratifying than anything I can possibly imagine. Second, the challenge of finding a new drug is incredible. We are trying to solve the complex disease with single molecule. It is incredible challenge. This is a big challenge, using a single molecule and finding out for various disease. We employ technologies that are just unbelievable in depth, their complexity. At the end of the day, we do this being some comfort to the people who are suffering and having dealing with anguish and desperate for chronic disease. It is there may be some hope. So we are trying to use lot of technology to develop a drug very fast, which is much more useful, and it may be a great contribution for the human mankind. I think this is my last slide. Thank you very much for your patience listening. Thank you very much.
Sir, thank you, sir, for sharing your knowledge with us. And our participants is also very grateful for you. And sir, you covered all the topics of preclinical, clinical, biomedical, whatever thing, artificial intelligence, all those things, sir. Thank you very much, sir. Sir, shall we have a small query session, sir, for our participants?
Definitely, sir. I'm very happy to answer that.
Sure, sir. So participants, you can raise your hand. You can directly ask the sir if any queries regarding this topic. Participants, you can raise the hand, or you can type. Sir, Dr. P. Dwarakanath Reddy, sir is going to ask some few queries regarding this concept. Sir, unlocking. Sir, you can ask, sir. You can ask the queries, sir. Dwarakanath, sir.
Sir, good afternoon, sir.
Good afternoon, sir.
Sir, what is the importance of PET scan for useful for COVID-19, sir? or patient?
You're telling PGP?
Yeah. How that
PGP.
scanning, that scan? PET.
Yeah, that scanning is very useful usually to detect a very early complication, because you are talking about the corona related.
Yes, sir.
Yeah, in case actually, if you generally know that most of the coronavirus affect actually the lung, and there is a significant amount of inflammation, and then there is a microvascular damage. And this microvascular damage actually cause very microthrombi into the blood vessels in the lungs. And this technology is very useful in picking up very early complication of lung. And if at that point you pick up and you treat, you can actually definitely bring the patient back. Because by the time you do CT scan and find out the score, lot of lungs are damaged. So all the technology which I am telling, they are very, very sensitive in picking up the effect in very early stage. That is the advantage as compared to actually other like X-ray or CT scan or MRI. When there is a frank damage, then only you pick up. But these technology which actually I showed you, they are very, very sensitive, and they can pick up even very early, very minute change, because they are highly sensitive, and most of them are non-invasive. That is the advantage.
Okay, sir. Thank you, sir, for saying the query. If anybody is there, participants, if anybody is there to ask the queries. Please raise your hand so that we can you can directly talk to the sir.
Normally, they say if there's no question, there are two reasons. Either they have understood everything, so there is no question, or they have not understood anything. So what question they will ask? So asking question is good.
Yes, sir. Yes, sir.
So I will be more happy to answer question, because many things, because of time limitation, I may not be able to cover, but definitely I can answer in a question answer form.
Sir, can I ask you one question, sir, please?
Please,
Sir,
please.
you said
Very
that
happy to.
chips model, sir, chip model.
Yeah.
So, if those chips can make an inflammation in that particular region or not, sir? That means if that chips can able to elicit some inflammatory reaction.
These chips are actually ready-made, sir. Depending on actually your requirement, these chips are made. So, for example, you wanted to do the study on some anti-inflammatory drug. The chips can be made which actually having a inflammation, and they are actually producing all inflammatory markers, and you can test your drug. So depending on your requirement, what actually I was talking is the organ on chip, which actually you can use for study a particular organ. For example, you want to know whether your drug is affecting kidney. Okay. In the form of improving the kidney function or causing damage. So these are actually organ on chip. But there are disease models also. Depending on that, actually one can develop that and one can use that, because these biomarkers are actually produced. Okay. Presently, these organ on chip are mostly used for very early screening of the molecule, whether they are active or they are causing any actually damage, mostly like actually liver damage, kidney damage, heart problem. Very quickly you can pick up with organ on chip. In very small dose you can find out. Suppose there is a cardiac arrhythmia, your drug is causing cardiac arrhythmia. You can just use organ on chip that is hard, and you can very clearly see that once you add your drug, there is significant cardiac arrhythmia, and you can actually take out your drug outside. You need not to do further development. So that is the advantage of that. For early detection of the toxicity and safety of our molecule, these are very, very good. Hepatotoxicity, cardiotoxicity, very quickly you can pick up. And they are very nicely correlating.
Yeah, okay, sir. Okay, sir. Thank you, sir.
But still these technologies are very very few centers actually in the world. I was lucky to go to the center where I have shown that the individual who got the prize on developing, I could see they were doing a lot of work, but still it is not popular. They are thinking that maybe coming 4 to 5 years, definitely is going to become very popular because it's very simple, very easy and very predictive.
Okay, sir. Okay, sir. Thank you, sir. Thank you. Thank you, sir.
Hello, sir.
Yeah. Yeah, please. Yes, please.
Good morning, sir. This is Dr. Satish Kumar
Yes, Satish, tell me.
from the Department of Pharmacology, Bapatla College of Pharmacy.
Welcome. Thank you very much for inviting me for this
Sir.
lecture.
Sir, we are very much happy and we are very much thankful to you.
Same is because I'm also very thankful that
You accepted our invitation and
something which we are
you
doing.
given a very detailed lecture covering the whole aspects of preclinical to clinical pharmacology.
Yeah. Are you having any question?
Hello.
Right.
Sir, can you hear me, sir?
Yeah, yeah. Yeah, I can hear you.
Yeah, yeah. The participants are mixed of chemistry and mixed of pharmacology as well as other disciplines of pharmacy as well, sir.
Yeah, yeah, yeah. Because you are actually the
I
subject.
take this opportunity to inform the participants that many of you are pharmacologists and Professor M.U.R. Naidu is one of the leading pharmacologists or eminent pharmacologists in India. So take this opportunity to interact with sir and not only regarding this particular topic, anything related to pharmacology from preclinical to clinical side, you can make use of sir's expertise and you can ask your questions directly. This is a wonderful platform. Make use of it. That's our request from the organizing team side. Sir, we'll just wait for 2 or 3 minutes so that
No problem, sir. I will be more happy to answer any questions.
And your voice is not audible, sir.
I don't. Maybe from your they have only put into mute. They have put into mute. I don't know.
Yes, sir. Yes, sir. Yes, sir. Now we are able to hear you.
Yeah, yeah. So they should not put on mute. They are putting mute every time.
Okay.
So what I mean to say that I will be very happy to answer any question today or subsequently. They can have my email ID from you and they can approach me.
Okay.
If anything
Sir,
is there.
Sir, meanwhile, when we wait for questions, we have Professor K.R.S. Sambasivarao, sir, who is the Vice Chancellor of Mizoram University, Central University, Mizoram. We're
Yeah,
happy to have you, sir.
he may not be knowing me, but I know him very well.
Okay. So
Yeah.
I think now you can mutually speak to each other and as well as you can address to the gathering as well, sir.
Yeah.
One moment, sir.
Good afternoon, sir. His speaker is muted. Mic is muted. He has to unmute.
Now I got it. Hello.
Yeah,
Good afternoon.
Good afternoon,
Good
sir.
afternoon, sir.
Thank you very much.
KRS Samsara, sir.
Yes, sir. Yes, sir. How are you, sir?
I'm fine, sir. How are you?
I'm fine. I'm fine.
Sir, good
Good,
afternoon,
good,
sir.
good.
Yes,
Yeah.
sir.
Sir, voice
Actually, I'm sorry I could not join because I'm traveling, sir.
Okay.
I just reached Delhi.
Okay.
Sir, did you hear me, sir? Sir, your voice is not hearing, sir.
Is it okay? Is it
I'm able to listen.
Hello? Is it okay?
I'm able to listen.
Yes, yes, it is okay. This is the maximum sound I get. Hello? Hello?
Hello, yes, sir.
Hello?
Sir. Sir, it is hearing, sir.
Yes, thank you. Thank you.
Sir, so dear participants, so first of all, I would like, it is a great honor for me to share with you the today's program, and I am delighted to have the opportunity to addressing the eminent personality in pharmacy, Dr. KRS Samsarao Garu, Vice Chancellor of Mizoram Central University, and he held many prestigious positions and several patents and awards in the fields of pharmacy and biotechnology. So he has 241 research papers, 8 patents, and also having 35 years of research experience in the fields of environmental toxicology and physiology, environmental, aquaculture biotechnology, fermentation technology, as well as pharmacy. And he's also a reviewer for many international and national journals. Under his esteemed guidance, 25 PhDs were awarded, and he is awarded with the Best Teacher Award from the AP government, and he is acting as a Vice President of Indian Society of Comparative Animal Physiologists. So we are very glad to be here, you sir.
Thank
sir,
you.
Just you can share your views regarding these program to our participants, sir.
Good afternoon, everyone. Respected the principal of the college, Professor T.G.K. Murthy Garu, and the committee members, Manam Nageswara Garu, and other members, other committee members, President of the college society, and B. Venkat Sivarama Krishna Garu, and P. Bhavan Narayana Chaudhary Garu, and C.H. Venkaya Garu, and other members, faculty, teachers, and participants. It is a great pleasure for me to be here because Bapatla Pharmacy College is one of the best colleges in Andhra Pradesh, and I always love to be there in physically, always in that college because of the facilities which they have, and no other pharmacy college in that area are having that kind of facilities which they have. And actually, I would have been physically been present in Bapatla today, but unfortunately, I have to travel to Delhi because of the sudden official assignment to Delhi. I could not travel, and I was just told by Murthy that I should be there for 5 to 10 minutes in the workshop, which is going to be started today, and also which was sanctioned by AICTE, and it is a refresher course program. And on in silico drug discovery and design methods and applications. It was a wonderful topic for pharmacy students, in silico drug design, because as you know that drug design, it takes place minimum 10 to 12 years. And for getting a drug, we need to go for several kinds of formulations and also clinical trials and also drug development and drug efficacy and drug biological activity ought to be tested. And after that, it should go for clinical trials in phase 1, phase 2, phase 3, phase 4. And then only the approvals from the concerned authorities, it is to be required. Then this process almost takes a period of 10 to 12 years. And it is a very tough process. And it is sometimes the drug which has been approved, that is found out as a very efficient drug by us, it will not be sometimes not been approved because of various reasons by the particular authorities. So it is very important that we need to have a drug which is effective in all the possibilities, and also it need to be confirmed that this drug is very efficient. So the in silico drug design will be useful, and there is no problem, sir. Kuchan, sir, no problem. And in silico drug design is a technique. You can have a screening of several drugs and several possibilities of the drugs you can arrive. And in silico drug design, you can have several possibilities by modifying the drugs which are existing in the present situation. And the a drug which is very effective for functioning of particular disease, control of the particular disease, you can plan. And this in silico drug design actually, which are not being present at present, because you are developing several drugs at a time, but they may not be effective when you are going for clinical trials and other things. So if you are going to have a in silico technology being implemented, this will be very useful to have a effective drug finally, and that can be taken to laboratory conditions. And this in silico to in vitro conditions, it is more important that how we are going to screen the various drugs, and how the drug efficacy is being tested, and how the drug is going to be affected target activity of the particular drug also can. Nowadays, available in the present market are going to develop resistance for various pathogenic organisms and also for various diseases. And it is UK universities and the several companies actually, they have a presumption that in the coming decade, all the drugs are going to develop resistance for the human beings. And in the future, all the synthetic drugs which are being developed are going to be ineffective. And this requires some kind of an alternative mechanism that we have to isolate the drug from the living organisms and biological organisms, and you have to isolate the secondary metabolites which are being produced by the plants and animals and the microbial organism. Those secondary metabolites has to be taken into consideration, and we need to develop several drugs. And even these compounds which are being isolated from the living organisms are not known to us whether they are effective, that the pharmacy students has to keep it in their mind. So these drugs which are actually effective, and even sometimes the existing drugs like you have ampicillin and you have anacin. Anacin is going to be used for since many years for headache, but nowadays anacin is not been effective for control of headache. Why it is? Because it is continuously being used by the same person for several years. So it is not been able to control the headache which we are getting. Similarly, penicillin, it is going to be used for several antibiotic symptoms, but it is not going to be effective in many patients. So why the penicillin? Because of the usage since many years, it has not been effective. The penicillin has to be modified at tertiary level or secondary level or primary level. It is to be modified to be an effective drug. This is one alternative. And the in silico mechanism will be very much useful to modify the drug which is already available, and it will provide you a effective drug. And other important point you have to keep in your mind that the recent development in pharmacogenomics is one of the area where we don't know which drug is going to be effective for the particular patient. And for this, if you go to a doctor, he will provide several medicines at a time. And those medicines which are going to be given for the particular patient, we don't know which medicine is going to be controlled for a particular disease. And doctor doesn't know that what disease actually the patient got. And it is a trial and error basis. Doctor is giving several medicines at a time. And this is one of the reasons that because many diseases which are coming in the human beings are genetic disorders. There are more than 5,000 genetic disorders. Now it has been gone up to 7,000 to 8,000 genetic disorders in the human being. And it is because of the single nucleotide change in a particular gene. ATGC pairing will be there. Even one nucleotide pairing is being mismatched. That is going to result in a particular SNP, single nucleotide polymorphism. These several single nucleotide polymorphisms which are been taking place, several of them may result into a particular disease. These genetic disorders has to be rectified through genetic rectification. But doctors doesn't know about that, and they will be using several medicines. What is that they have to do is particular genetic analysis of the particular individual has to be done. So nowadays, this pharmacogenomics is one of the area that is pharmacy and molecular biology combined together. And this area, they will be doing your genetic analysis, and particular SNP will be identified, and treatment is being done. And we don't need to go for clinical trials of the drugs for 12 years now. If we are going to have the genetic analysis, individual target-based drug can be developed now. Within 1 year period, you can target the particular group of SNP individuals and develop the drug. This is what one of the beauty of the drug development. So these are the areas need to be taught in this particular refresher course. And these are the areas which are actually has to be given emphasis, particularly for the pharmacy students. And this will be definitely going to be very useful to the young faculty members, and they have to concentrate on the research areas of these things. First one is how we are going to reduce the drug formulations, drug development, and drug efficacy, and drug biological activity. And second one is how to isolate a very effective drug from the biological organisms living like plants, animals, microbial organisms. And the third one is the identification of the genetic diseases and single nucleotide polymorphism identification, and drug targets has to be developed, and individual based drug has to be developed. And fourth one is very important one is actually you need to concentrate on the drug which is going to be very effective for a particular patient has to be developed in a particular individual group of people. For example, one drug which is going to be effective in your human body, it is not been effective in the next person because of various reasons. Drug metabolism and physiology of the drug also very important. So this group of individuals who has got the same similar single nucleotide polymorphism and group of individuals also can be identified. Now this is in Europe and North America. All the people actually are following this drug analysis, and also genome analysis, and drugs are being developed. So with this few remarks, the workshop which is going to be started today, inaugurated today, and AICTE workshop which has been sanctioned by AICTE. I congratulate all the faculty members of the Bapatla Pharmacy College and the College of Pharmacy Bapatla, and all the principal and other management members for providing an opportunity for me to interact with all of you. I'm sorry I could not spend much time. In the middle I joined and I could not inaugurate the program, but I'm always there, and if it is required I can join again and give a lecture, because I will be here in 3 days. After 3 days I can join and give a lecture on this either in the pharmacogenomics area or in the drug development area or in other areas. Actually I can also think about. There is one more point actually I wanted to emphasize, request the college principal and other faculty members. There is another point, another possibility for drug to be ineffective when it has been isolated from the microbial organism, because the post-translational modifications of the drugs which are being synthesized from the secondary metabolites are going to be one of the key factors for development of the drugs. Because when you isolate the secondary metabolites from the plants and animals and microbial organisms, they are not going to be very effective in the same manner how they are when they are isolated from the human cells. So this is also one of the post-translational modifications. The carbon-carbon interactions are also going to play a crucial role. This particular aspect also has to emphasize for teaching to the faculty members and students during this workshop. And if these are the areas actually in in-silico drug development, very important areas, and you can concentrate on these things and you can teach them. After 4 or 5 days, if necessary, I can even come down again to Bapatla and spend some time during my stay at Guntur. After 9th, 10th, 11th, I will be there in Guntur. I can also think about coming there. Thank you very much, Mutigaru and other faculty members, for giving me this opportunity. Okay.
Thank you very much, sir. Sir, just a moment, sir. Principal sir is coming here.
Yes, yes. If students has got any doubts, they can ask me. When this, I think 1 week refresh course or 2 weeks refresh course.
It's a 1 week program, sir.
Okay, okay. In some of the areas which I mentioned, if nobody is teaching, I can come back on 9th and I can even give an online lecture if it is required.
Sure, sir. 9th, sir.
I will be coming back to Guntur on 9th.
Okay, sir. Okay. So I request all the participants that we have both Professor M.U.R. Naidu Garu as well as Professor Sambasiva Rao Garu, both of them available, and make use of this opportunity. And if you have any specific questions related to pharmacy, pharmacology, or anything related to pharmacy, you can use the raise the hands option in the Zoom platform. And as well as if you have any other queries, you can always speak to the both the guests.
Good afternoon, Honorable.
No problem. I can talk to Murthy later on.
Okay,
I think he
sir.
has got internet connection problem.
No, sir, I am here. Hey, good afternoon, Samsara Garu and Naidu Garu and
Good
other.
afternoon, sir. Good afternoon.
Good afternoon. Thank you very much, sir, for sharing your valuable time and so that it will be useful for all the participants. And in spite of hectic schedule, Professor Samsara Garu graced this occasion. And on behalf of the management, faculty, I wish to extend my wholehearted thanks to all of you so that the faculty members who attended this online faculty development program will have a good benefit. Thank you once again, all the resource persons.
Hello?
Hello?
Hello? Hello?
Hello?
Hello?
Hello? Samsara Garu.
I think I have internet problem, I think.
Okay, sir. Okay, sir. I will talk to you later then.
Sir, क्या 9 बजे का करा दूं सर?
Yes, sir. Anybody joined for lunch from the college also? The coordinator is coming? Because we have to talk about the program of tomorrow and after tomorrow.
Vice Chancellor will come, sir.
And then you can ask quiz coordinator also because we have to, at the time of that, no, even she can come at 8 o'clock. No problem. Yes, sir.
Okay, good afternoon, Samsara Agar. Now you are having some net problem is there, I think. Naidu sir.
Yes,
Are you
sir.
able to hear, sir? Dr. Naidu Garu.
Yes, sir. Tell me.
Okay, sir. Thank you very much, sir. And we are waiting for day after tomorrow's session.
Yeah.
Very much useful to the participants.
Yeah.
Good feedback I received.
Yeah.
And definitely it will be useful to the faculty members and they will be eagerly watching for day after tomorrow's session, sir. Exactly 11 o'clock.
Okay.
On behalf of this management, once again, I am very much thankful to you, sir.
I am also very much thankful for you to giving me this opportunity to interact and talk to people. And really, very nice. This thing is there. So I hope that I will definitely will be on day after tomorrow. I'm going to talk on something much more advanced and very interesting on drug discovery in the post-genomic era. So
because the younger generation need your blessings, sir.
Always there, sir.
Thank you, sir.
Okay.
Have a good day, sir.
Right. Good day, sir. Thank you very much.
Thank you, sir.
Yeah.
Now the session will be handed over to Sudhir, the coordinator.
Okay. Thank you very much.
Thank you, sir, for spending your valuable time with us.
Yeah.
Thank you, sir.
So.
Okay, sir.